Fluorescence. In this picture, there are many mice. Can you find the mouse with a different shape? Well, if you have a keen eyes, you can find this mouse has a bigger eye and another one here too. If the mouse has a color like a red eyes like this, then it's much easier to us because human being, we have the ability to recognize a different color. However, if we turn off the light, even though this mouse has a red eye, we cannot see the color. That is because this color is a subtractive color. Subtractive color means this color is generated by absorption of other color and only red color is reflected to our eyes. So in this case, if the mouse eye can generate light by itself, it could be much easier to see them. Also, it would be much more sensitive. This kind of a phenomena is called luminescence. Luminescence is generating light, and light is a highly condensed form of energy. So that's why to make light, it requires the strong input energy. Depending on the input energy form, it can be classified as a bioluminescence or chemiluminescence. If it is electricity, we can call it electroluminescence. Sometimes the input energy can be also shorter wavelengths of light. In the case, we call the phenomena as fluorescence. Firefly is an example of bioluminescence. Utilizing ATP energy, it can generate the visible light like this. The chemiluminescence examples we can find from glow stick. In glow stick, hydrogen peroxide has been used for chemical energy source, and it can generate the energetically unstable intermediate, and it can be converted into chemical luminescence. Electroluminescence we see in daily life, and conventionally we have used the fluorescence light, but these days LED is a more common and more efficient light source. In fluorescence, the input energy is light. And under daily light, it looks like just a white stone. But under UV light, the observed UV light converts that energy into visible light. This kind of fluorescence is not only in the mineral, it's also found in many different forms of life. And these are different from bioluminescence. These materials absorb UV light and then change that energy into longer visible light. For generating fluorescence, first of all, light should be observed. And then, by releasing some part of the energy, and the excited molecule is aligned here, and then it can emit light. Because the emission light is smaller amount of energy than observed light, usually the wavelength is shift from absorption to emission, becoming longer wavelengths. So difference between absorption light and also emission light, the wavelength difference is called as a Stokes shift. The absorption of the molecule can be defined by this formula, and absorbance is the extension coefficient multiplied by band path and also multiplied by concentrations. So amount of the material, the concentration is high, then absorption is also increased. And band path is increased, also it can absorb more light. But most importantly, the intrinsic property of extension coefficient, this should be high, then it can absorb more light. The brightness of the molecule is defined by extension coefficient multiplied by quantum yield. How do we make the dye molecule into fluorescent molecules? If the dye has a rotating bond, then usually the observed light can be lost by mechanical heat generation. but if you combine those molecules by this kind of a bridge and then suppress the rotation, then this molecule becoming more rigid. Then this molecule has a better chance to keep the energy and then emit those stored energy as light. So this is the common mechanism how to convert regular dye into fluorescence dye. Here are the representative fluorescent dyes. Y-axis shows the brightness, and then the high number here means that these are bright dyes. And then x-axis present the wavelengths. So here are blue color and green, yellow, red, and long red colors. Here are the most representative fluorescence dyes in visible light. Blue color dyes is DePi and Haste are most important nucleus dyes. Green color is represented by fluorescein. Fluorescein isocyanate, abbreviated into Fc, this should be specific compound name like this. And chemical structure for the fluorescence motif should be fluorescein, but by the biologist, Fc is kind of a represented name for the dye and also the filter set of microscope. Orange color dye is a TMR, tetramethylrhodamine, and red color is a Texas red. Both of them are rhodamine structure, but with a more ring structures. Texas red is near to 600 the nanometer wavelengths. Cyanine dyes are also very important and famous the dyes because they have a very strong extension coefficient. So size 3, size 5, size 7, these are the representative cyanine dyes. Here the number 3 is the counting the bridge molecules. 1, 2, 3 carbon is a size 3. 1, 2, 3, 4, 5 is a size 5. And then 7 carbons in between, that's a size 7.